to the Weekly Bioanalysis, a KCAS podcast.
Hello and welcome to the 76th episode of the Weekly Bioanalysis, the official podcast of KCAS Bio. KCAS Bio is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. My name is John Perkins and I've been in the bioanalytical LCMS space for over 29 years, and I'm here with my co-host Dominic Guarino.
Hello, my name is Dominic Guarino and I've been in the LBA and cell and gene therapy space for close to 30 years now.
Dom and I are members of the growing scientific advisory team at KCAS. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS's services. We are thrilled to have you listening to the 76th episode of the podcast, which is now available virtually everywhere. Wherever you choose to find and play your podcast, you can now likely find the Weekly Bioanalysis. We welcome all of you, whether you are joining us for the first time or if you're a regular listener. Today's podcast will be a review of the latest news and resources, and then the focus on a topic of our choosing before discussing any feedback we've had from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. So again, we're amped to have you here. No, we're not going to. I'll do that again. So again, we're excited to have you here, and we look forward to a fun episode today. To kick us off for podcast number 76, Dom is going to go over today's podcast topic a bit before we jump into the news and resources section. Dominic.
John, we're fired up today for GLP-1 Analogues. That's going to be what our main portion is, and some of these blockbuster drugs with multiple applications in the bioanalytical challenges. First, we're going to go over a little bit of the mechanism of action of GLP-1s, some of the roles of GLP-1 analogues in diabetic treatment. We'll talk about the structures. Maybe we'll talk a little bit about how they're used in weight loss. Of course, that's all the rage. But then, John, supply chain issues, some of the bioanalytical challenges, we'll really dig in there a little bit, and also some of the future of GLP-1 analogues. It's a jam-packed show, John. Really an interesting, fascinating topic. Excited to talk to you about it. All right, John, as always, let's just jump right into the news and notes here. We got some interesting articles to go over today. Tell us a little bit about what Vertex Pharma is doing.
Yeah, so I don't usually focus on particular companies, though I think I've done it a couple of times today. But this is interesting because it's actually, the headline is Vertex Pharma scientist talks about a long road to developing non-addictive painkillers. With a supplement to that, the Vertex inches closer to acute pain market as FDA accepts VX548, and this is basically Vertex. Well, it comes down to how do we address pain. As the scientist who was interviewed for this, who is Vertex's chief scientist, Dr. David Altshuler, talked about Vertex's research and development plans, really focusing on this VX548. But what we have in terms of our tools for pain are things like Tylenol, which are moderately efficient but well tolerated. But then the other camp on that are things like your opioid analgesics, which are very effective, but of course have the possibility of addiction. So Vertex have actually taken a completely different approach in terms of how they're looking at pain, in that quest to see if they can figure out how to develop, or if they could figure out how to develop a non-addictive pain medication that was effective. So the really interesting background to this is they've been working on it for 20 years, and the actual insight that led to the medications developed actually came from studies of people who had a rare condition where they were insensitive to pain. They can feel things, sense temperature, but do not feel pain. And this was actually identified in a family of firewalkers who could walk on hot coals. So in terms of looking at this family, they figured out the condition was due to inherited differences in a particular protein that has a role in pain signaling. So if you lack this function, you don't feel pain. So in his words, we and many others have worked for decades to make a medicine that could recapitulate that naturally occurring phenomenon. The article doesn't say how that's all addressed, but ultimately that's what so that's what drove the direction they went in terms of developing this medication. They've had it in, I think, phase 3 trials. It's shown that it can definitely reduce pain. I think in one phase 3 study it didn't match opioids, but then it still has the benefit that it's non-addictive. So so if it might not be quite as good as opioids, but if you're better than Tylenol, etc. But you're like I say, you're not, you don't have the addictive properties of opioids. Maybe you're in that middle ground, or better than middle ground, that actually makes this a drug that really could have legs. And that's why I wanted to talk about this.
Yeah, no, John, it's a fascinating topic in general. Just like trying to take any sort of drug that can give you a side effect of getting you high, so to speak, and then can it have medical purposes like cannabis and things like that. So the opioid thing is no different here. And this was, I didn't realize people had started 20 years ago on this. That's great. The piece that is always interesting is the history around it. I think of it as the etiology of it, right? This came from somebody who was like, hey, if I could get these firewalkers over here and figure out what they're doing. And, there are people who are asymptomatic to aspirin, right? There are people sometimes they're given these drugs and they're like, no, I feel fine. Ouch, I can still feel that. So that's still some unlocked things. There's even like for years, not for years, for maybe 6 months of my life, I spent trying to look at a uniflow test to look at like a pinprick of the finger to see if you were asymptomatic to aspirin, John. Don't ask. Yeah. So it was a holy grail of science and not something that was ever really probably achievable in that setting. And actually I was helping out somebody trying to fix something on there. So it's kind of a convoluted story, but anyhow, the reason why I bring all this up is there's a Netflix series called Painkillers. It's fascinating about the whole Purdue Pharma incident, and that spawned this. A lot of it's generated from that. It was a part, we remember, we kind of, at least I say grow up through it. We lived through it, right? It's still a problem.
Sure. Oh, absolutely. Yeah.
And everything like that. But the opioid issue, I mean, they called them drugstore cowboys, right, John? Like it was, that's why you see things locked up. So this has been something that, they're breaking through on it because these are powerful things, especially as I start to senesce and get older. I start thinking about, wow, what is it like to have some of these things, or what might it be? So this is great stuff, John. I don't know. I don't have much to add. I don't know too much about like, there's some pain biomarkers we've worked in. So there are some biomarkers in this space, but as I mentioned, there's no real test.
Yeah,
I'm just going to put people on firewalker, right? You can't make them walk across fire, but there's no test for this type of stuff. So go ahead, jump in. Yeah,
just going to add a couple of things. It's one of the questions within this: why wouldn't these drugs carry the same addictive properties as opioids? And basically, it's a different mechanism. So the opioids are working in the brain, whereas he doesn't talk about the mechanism of action. But the mechanism of action here is actually on the periphery of the body. Not in the brain, so it's coming from a complete different process, and that's the way it's not shouldn't be an addictive. It shouldn't be an issue of addiction. And in terms of other studies, they're also looking at diabetic peripheral neuropathy. So that's in phase 2 and looking forward to phase 3. And then they also have a phase 2 trial looking to start with lumbosacral radiculopathy, which is caused by inflammation or irritation of nerve roots in the lower back. So, I mean, think about back pain. I'm hoping this one gets to the market.
Yeah. You don't want to be taking Vicodin, right? You don't want to be chewing Vicodin there all morning long like people do. Yeah.
And that's really interesting because as many adults are, I'm prone to back trouble, and just every so often it can get triggered, and it can be really debilitating. But I honestly found that when I was given oxycodone, I saw no effect of it, so I actually discontinued it quite quickly because it didn't bother me at all. I actually found that Advil was more effective for making me comfortable and keeping my back pain under control. Whereas my wife, when she has migraines, will do a mix of like Tylenol and codeine to help with that, and then it's a very short burst of treatment. But yeah, it's like I say, it's one of those medicines that'll be one to watch to see if it makes it to market, and then where it goes from there. And it has actually moved from, it was started as VX548, it's now called suzetrigine, so it has a name, and it's something to watch out for. So
Yeah,
yes,
no, it's great. I think it's a really germane topic. You're going to see more and more people living longer. So therefore, by default, that can come with neuropathy, not just diabetic, right? So back pain, all of it. We're body biochemistry. And hey, I'm a big fan. If I could take something that, John, unlike you give me an aspirin and I'm wound up. Like this stuff has a real effect on me. I've only been prescribed. I had my wisdom teeth removed when I was 16, and I think I got some like they were Rockasets or whatever it might be. And I just remember that was a lot. And maybe one other time for another small outpatient surgery where I got prescribed something, and my wife's like, "You okay?" I'm like, "I'm feeling great." So this stuff, it has an effect on me for sure.
yes, yeah, it's
Yeah.
yes, they were all different. That's the nice thing. So
Yeah,
that's what
any of them, all of them. Like if I have any moderate, sometimes like I've been running lately, I might take a small, like I take one aspirin. It's a running joke. Even my 18-year-old looks at me, is like, "You gonna be okay taking that?" I'm like, "Yeah, shut up," So because it does get to me. So I don't know if you want to. You kind of covered the Veritex thing. They're a
yeah,
Boston
that's all
company,
one
great
and the same.
company.
It was two
Yeah,
articles covering the same
awesome.
thing.
Let's keep
So
rolling
so the
along.
next thing is just a quick bit. I thought it would be interesting. Again, it's focusing on a particular company, but it's Prime Medicine receives FDA clearance to run a first prime editing clinical trial, and so this is the first ever human trial of prime editing, opening the door for testing new technologies that backers hope will widen the array of diseases that can be treated with gene editing. And I think in the past, the FDA has been fairly resistant to, or has been resistant to this kind of approach, or in terms of clinical studies. But like I say, this is first of. I don't know, but sorry. Let me back up on that.
Mhm.
The basically this therapy is a treatment for chronic granulomato- granulomatous disease, CJD, a rare genetic condition in which phagocytes um, don't work properly. Typically diagnosed in early childhood, it can cause people to get seriously ill from bacteria or fungi that would normally cause few or no symptoms. It's caused by mutations to the genes encoding for a key enzyme called the NADPH oxidase that white blood cells need to fend off infections, and their treatment is meant to correct the gene mutated in one of the variants of CGD. So basically, the treatment involves extracting blood stem cells from the patient and sending them to a lab where prime editing would then be used with the goal of correcting the mutation. Cells would then be infused back into the patient, hopefully to restore immune functioning. So basically prime editing is looking to rewrite short segments of DNA. It doesn't require double strand breaks as with CRISPR-Cas9. It's out of David Liu's lab at the Broad Institute of MIT and Harvard, and is only a recent development. So it's the background to this is the FDA really has been slow or
Yeah,
cautious about moving in this direction, or clearing human studies of new gene editing treatments.
yeah,
And this was only rolled out in 2019, so this really is a new approach.
yeah.
So you can understand their conservatism here. So I just thought it's interesting because it's first of a kind, and again, who knows, it may go nowhere, but it's that first step and onwards that probably will mean it's a direction that will acquire legs in the future if this doesn't go anywhere. And that, it's another medical approach we'll be watching out for.
Yeah, no, John, it's a lot to unpack here, right? So we're for me, it's I'm back in 1994 where I'm doing my first ex vivo autologous therapy, and here we are talking about it again, right? And
Oh, yeah,
it's
yeah, yeah.
so far, right? I'm going the other way. It's actually evolved. This is just like another evolution in the ex vivo therapy space, and you touched on it about the current CRISPR technologies. Um, they have this requirement to break double-stranded DNA, which in and of itself is just dangerous, right? That's what carcinogens do.
Mhm.
That's how breaking double-stranded DNA is how you kind of kill a cell or mutate the cell. So but they're now finding a way. And by the way, those initial CRISPR technologies, they did have some there was some fear of side effects. They've engineered them in such ways to prevent them, and this is that evolution. This is the next engineering. So they've unpacked a way to rewrite those short segments without having to break it. They don't really talk about it here, but that's their IP, right? And David Liu, I mean, we need to have this. We gotta try and get him on the podcast because he's, here he is again. I mean, you couldn't be a bigger name in this space than him. And this Prime Medicines looks destined to do some great things. But these types of evolutions are, this is why we love the field, John, because it takes years in the making, and this one kind of popped up pretty quickly. I'd say the CRISPR technologies themselves have been one of the faster moving, and I think the CAR T cell space kind of carved the way, right? Because those technologies, I mentioned it in the middle part of the 90s, started realizing this is powerful stuff. How do we, there's all sorts of, I won't go over, it's like its own little probably podcast, but basically it took decades before the first CAR T cell got approved. And even now, as there's still a bolus of them that were recently approved, but now we've got, there's some caution here because this kind of safety thing, and they'll start to, we've talked about that, they'll mitigate that, right? So, but this has allowed the CRISPR, what we've learned from all that it's still an autologous, they're both autologous ex vivo therapy.
Mm-hmm.
Right?
Yes, exactly.
And so that's the piece that allows this it's leveraging that. So it's made leaps and bounds, John. When we started this podcast, I don't know, 4 years ago now, I would have said we're a decade away. So they've cut it in half. You know what I mean? I really do. This is me, I'd take this if it came to it. That's what I'm saying. That's how advanced it's been. Where if you had asked me 5 years ago, I might have been a little more like, I better really be sick, right? But now it's like, hey, if I think these things have, especially as I read this. So I'll stop there, John. Thanks. This stuff I love, John, right? I love.
Yeah, so again, like I said, it was just a highlight of where the industry's going and things to watch out for in the long term. The next piece is, this is quite early work, but it's gut enzymes could make universal donor blood possible. So a completely different direction.
Yeah, John, huge thing too. Another holy grail of science.
Yeah, absolutely. Yeah, it's really so. It's funny when in terms of news articles, it's actually been quite light, but there's still some real gems in terms of what people are doing. And
well said. There's some activity out there, but these are some of the things that have come out are huge. Like they're like, wow, this is
yeah, sure, exactly. So this is actually a couple of European teams, in an article published in Nature Microbiology. A team of researchers from the Technical University of Denmark, DTU, and Lund University in Sweden detailed how they discovered gut bacteria enzymes that remove not only A and B antigens from red blood cells, but other components that have historically hampered progress towards universal donor blood. So they say we're close to being able to produce universal blood from Group B donors, whereas there's still work to be done for the more complex Group A blood, and this was led by Maher Abu Hachem, a gut bacteria researcher and one of the study's senior authors. Our focus is now to investigate in detail if there are additional obstacles and how we can improve our enzymes to reach the ultimate goal of universal blood production. So I'm a non-biologist, so I actually didn't know why it was called O and B and A. So this was really interesting to me. That's how rudimentary my biology education is.
Well, don't question me, John, but I know a little bit to be dangerous here.
If you don't, I'm going to read this because it's key to the whole thing, and actually,
go ahead. I got the thoughts.
It really leads to the elegance of it. It's like blood donations must either be matched by type or come from a recipient with the only natural occurring universal blood donor type, O. Mismatched donations can lead to what's known as an acute hemolytic reaction, where the immune system attacks the transfused red blood cells. This was all figured out about 40 years ago. Scientists discovered a set of gut enzymes capable of removing antigens that characterize type B blood cells. A different set of antigens characterize A blood cells. Cells with both A and B antigens are type AB, while those without either of them are type O. So what the team then focused on is if they could remove the antigens, then they could make the blood similar to type O. And so let's see. yeah, I'll pass it back to you, then I'll
Yeah,
come
well,
back.
there's a couple things. Red blood cells are non-nucleated cells. They're tiny. Now you're going to treat them with some enzyme to knock this antigen type off of it to make them less immunogenic. That's basically what they're doing. So you kind of described it. If you're type A, type B, type AB,
Sure.
or type O. If you type A, you have A. If you type B, you have B. If you type A and B, you have both antigens. You type O, you lack them, right? So that there's a clear path to removing one of them. They kind of talk about, I think it's the A to B path, but all of this is it's just fascinating how they continue to like take parts that are in us all and figuring out how to repurpose them to do therapies. That's what I'm hearing, right? That's kind of cool, right? I say parts or enzymes.
And if you don't mind me jumping in, but and still things that we think we know about, they're still finding new information about them, because what led to this is one of the researchers at Lund, called Jennifer Ricci Hagman, identified a previously undiscovered extension of the type B antigen. And so, I mean, this is recent stuff, but we've been talking about this stuff like originally from 40 years ago. It's fascinating how we have these bodies, but we're still learning about them.
Like, the thing. See, that's why I said I know enough to be dangerous. I know this is dense, but they teach it to you like, oh, it's A, it's B, it's really easy, like I described it, right? But they just even in this article, 24 different types of A. So it's pretty cool.
And like they said, the finding of that extension on B then led them to look back at A and found there's similar extensions to that, and they think and similar extensions which, despite knowledge of their existence, hadn't been thought to play a role in blood type compatibility. So that's when the labs collaborated to sort of to use a bacteria to then take off not just the antigens but the extensions, and that's where hopefully your path to universal blood.
Good luck to them, John. It just seems so like a red blood cell so fragile that you're doing. If they still contain their, efficacy, I guess I can't. You know what I mean? But I guess it does. Obviously, I'm not quite. I'm no like red blood cell transfusion expert, right? I'm obviously. I'm more interested in what the T cells might do when they get in you. I've never been too involved in just straight up blood transfusions but and most blood products that we use, you're just throwing away the red blood cells or lysing them, getting them out of there. So, but they're something that obviously I've studied enough about, but these types of therapeutics just continue to fascinate me around how they,
Sure.
And the where I was going is, one of the best things, I had some great mentors and one of them said, hey, if you really want to know immunology, go study developmental biology. And I was like, what are you talking about? I'm like, why? But if you can unlock how a mother allows the fetus to grow, John, that's what a lot of it's here. If you could unlock those keys, and man, that's something that is, if someone said there's not an understudied area, but that's probably an area that has a lot of like value to try to unlock the keys of immunology. Any immune privileged areas are some of the best things to study. And your eye is one, the brain is one, and then of course the mother's womb is one. So anyway, John, it makes my mind go there. It makes me think about these things, and just any young scientist or scientists in general, oftentimes the key's just sitting right in front of you, John. Right? A lot of everything, almost every drug we talk about has some form of it in nature that we're just trying to replicate or isolate. And I think the sky's the limit. I still don't think we know all the receptors on cells, let alone red blood cells, as evident by you said it, 40 years we discovered these A antigens, and now even within the last year, someone discovered a new one. It's just, It's mind blowing, right? And then like everything we study is somewhat static. And when I say static, like the surface of a cell is like, that's like, it's just constantly up and down, moving, very dynamic. And then the gene area, I'm going to go deep on you, John. We still haven't unlocked all of the information we got from there and how it translates into proteins and surface.
Sure. Yeah, absolutely. Yeah. Yeah. So, it's interesting. These, like I say, these enzymes are from a bacteria which lives off. Bacteria is Akkermansia muciniphila, which lives off the mucus lining of the surface of the gut, and it could break down carbohydrate extensions of both type A and type B antigens. And that's where your 24 comes in. There's 24 different types of these enzymes from this bacterium, bacteria, and then they found that 7 were actually possibly capable of taking off sufficient to move to this more what they're calling ABO universal blood.
Yeah.
They're actually now they've patented the enzymes and the protocol for converting them, and the hope is that this will no long term be available for get to the clinic and be useful. So yeah,
Yeah, John, I'll close with one thing. I've worked on some platelet-like synthetic platelets, synthetic red blood cells. Great stuff. A lot of Department of Defense work. Incredibly powerful stuff.
yeah. So I thought it was, like I say, fascinating. I learned a lot just in that one small article. Let's move on to the main topic, since there's a fair amount to talk on that. So I think I put so basically I called this GLP-1 Analogues, Blockbuster drugs with multiple applications in a bioanalytical challenge. We've the GLP-1 analogues and the sort of tension between diabetes treatment and obesity treatment have been in the news for a long time, and it's sort of driven by celebrity endorsements and all sorts of stuff. And we've sort of just been watching this space without necessarily feeling it was a time to talk about it. We have worked on a number of these, and our non-GLP team have supported some work, and we hope to have them in one of our near future podcasts, and maybe we'll discuss this with them. But it just seemed that there was enough going on with the GLP-1 analogues to sort of dig in a bit into the background, why they're effective, so just some of the, and actually it was also brought to the fore because there's an article in, I think, American Association for the Advancement of Science, which is a really good review article, which it said innovators who fought to unlock GLP-1 drugs for obesity awarded the Manny L. Bolmick Breakthrough of the Year award. And like I said, it's a great article, and it just seemed like a good time for us to touch on this, particularly as the bioanalysis, they're non-routine is the best way to put it. They're I would say bioanalytically they're probably fun things to work with, but they're not really, because it's like instrumentally they're a pain in the neck. But we'll touch on that later in the topic. So yeah, that's the main topic. Let's get going.
Yeah, no, John, that's a good intro. I'll just jump in real fast to maybe.
Sure.
You're talking about oligos, right? And that's what makes them.
No. These are peptides.
Well, my mistake. So peptides.
Shall I give a little bit of the history before we?
I don't know why my brain slipped into oligos. My mistake. Peptides.
Because we spend so much time talking about oligos at the moment.
Let's take a moment, and they're GLP-1 peptides, right? And you can give us a big bolus of it, and I'll let you go in a second here. But these are bioanalytical challenges because of their size, right? They fall into that tweener space, right?
Yeah.
And so you have to do things, and you can drill into it. You can do things on the mass spec side that the mass spec doesn't always like. And then for us on the LBA side, the piece of real estate's so small, right, to begin with. And the part is there's a lot of homologies between these. Like sometimes it's just a single amino acid difference, right? And you might drill into that a little bit. But like there's a lot of it's tough to get an antibody that's very specific against, say, the drug or the endogenous version of it, or even in general to capture all of it. It has all sorts of challenges to it. So that's my little bioanalytical piece. But John, I'm going to put myself on mute, and you tell us all about this. You love this. Okay, go ahead.
Well, it's like I say, it's fascinating. I'll give a little bit about mechanism of action again as the non-biologist, but I think it's relatively. It basically GLP-1 is glucagon-like peptide. It's a hormone that stimulates insulin secretion after you eat. It helps you feel full and regulates your blood sugar. And this is where the initial interest came for it, was for people with type 2 diabetes, where they're obviously their hormone release and response can be ineffective or absent, and that's why diabetes leads to to higher blood sugar levels. So these drugs are they're really GLP-1 agonists, and they work by activating the GLP-1 receptor. They slow gastric emptying, inhibit the release of glucagon, and stimulate insulin production, therefore reducing hyperglycemia in people with type 2 diabetes. They also reduce feed intake and therefore body weight, making them an effective treatment for obesity. GLP-1 events its main effect by stimulating glucose-dependent insulin release from pancreatic islets. It's also been shown to slow gastric emptying, inhibit inappropriate post-meal glucagon release, and reduce food intake. So a lot of that is the reason why it works in multiple areas. And really, the like I say, the focus for these was very much diabetes, but it's similar to Viagra. That was developed. That was developed in terms of it was for a cardiac indication, but then
Sure.
they noticed side effects, and they I don't think those side effects were expected, but then they could see a market for it. Whereas with these, it's actually inherent part of the mechanism is the fact that it curbs your appetite, so therefore there's almost immediately a secondary market because beyond diabetes. The big deal with these, the first one on the market, was I think it was exenatide, but was marketed as Byetta, and I think the company was Amylin, who are a Southern California based company, and it was actually the Byetta was structurally similar to a protein or peptide that was found in the saliva of the Gila monster. So I love the learning from nature piece. The big challenge though with GLP-1 or the natural GLP-1 is that it doesn't hang around long in the body. And so it has an effect, but it's gone. So if you're trying to design a medication, then it's actually something has to be done to actually help them hang around longer. And this is where a lot of this AAS article comes in. It's talking to Lotte talking about Lotte Bjerre-Knudsen, who I think she was a key scientist at Novo Nordisk, and Novo Nordisk we'll talk about a lot because semaglutide, and then the other person whose name escapes me right now, who was affiliated with Lilly, but actually it was Lotte Bjerre-Knudsen who postulated that by adding fatty acid chains to these peptides, you could extend the half-life. And the first one that she, I think it was gelaglutide, was the first example of this. And I'll keep throwing all these different names out, but it basically it showed the concept of the half-life could be extended by adding the fatty acid chain onto the peptide. This is at the root of all these drugs. So semaglutide has a different length of fatty acid chain. Tirzepatide, which is Mounjaro, which is the Lilly version, has another different, again, non-polar chain added to the basic peptide. And you're right, there is a lot of commonality between the peptide at the root of it all, and so that actually could play into some analytical approaches, because we talked to Don and the hybrid team about that. But so that was the root of basically developing drugs that could then be successfully administered into humans because they're useful half-life, and they obviously had a good clinical effect.
Yeah, I think the half-life piece is really the key.
Yes.
What am I trying to say? These come in many flavors, right? The delivery of the GLP-1 is some technology too, John. Correct me if I'm wrong.
Yes,
Not just
absolutely.
So maybe I don't know where you want to go, but like when you kept talking about those chains, I thought, what does he mean by that? Like, I think I know what you mean, but maybe elaborate a little bit on
So
that.
a fatty acid is yeah, I'm supposedly a chemist. Well, it's really so a fatty acid is so when you think of a fat, it's basically it's a long
Positive
non-polar.
end, negative end, right? It's got
So
a
no, it doesn't. Well, a fat wouldn't. Well, when you say a fatty acid, that is basically a long. It's a long non-polar chain. So it's long. When we talk about non-polar, it's groups that aren't. Yeah, they're sort of they're I'm trying to think how to describe it in they're sort of it's sort of neutral to end. It's neither acidic nor basic. It's sort of neutral to what's going on. With a fatty acid, you've got this long neutral chain, but you have a acid group at the end of it.
Mhm.
And so that's going to be your route of attachment onto the peptide. So then, what the net effect is, you have then the peptide,
Okay.
they're linked through the polar group, and then you've got this long non-polar chain. It's like soap is basically like a long grease, but with a polar piece. That's how you.
Yeah,
Know,
and I
that's
think of
how you,
fatty acids and everything I've learned about what you're talking about is through like the cell layer, right? So I'm kind of
sure, yeah.
and like fatty acids have a wide array of the one of the lower number within the lipid bilayer, right? So but those things play a role and stuff. So anyhow, so you basically just throwing a fatty acid chain onto a peptide, right?
Yeah,
That's
you're attaching it. It's
yeah.
chemically bonded. So it
And
completely
do they wrap
changes.
them into LNPs and stuff, or am I mistaken? Don't some of these?
No, I think I think it's mainly just diffusion.
Okay, they
It's
just
yeah,
put this fatty
the,
acid chain
they,
onto this peptide. It's a hormone, right? Let's call it what it is,
yes,
right?
yes, but
And so then it just and that stabilizes it in the periphery
yeah,
and increases its half life.
but
That
certainly they're not well. There is actually some work being done on an
okay.
oral dose of semaglutide. So
I remember reading about that a little bit. No,
let
that's
me
good.
back up a bit.
Continue. Yeah,
Let me
continue.
the first one that Lotte Bjorn Knudsen came up with was actually liraglutide. We've worked on liraglutide. It's not dolaglutide, and there's so then Wegovy and Ozempic, that is semaglutide, and I think Wegovy, I think is that Wegovy will be the diabetic medication. Ozempic will be the anti-obesity.
Okay. So they are slightly different from each other, right?
No, I think they're just different
It's
dose.
branding, yeah, exactly. Okay, so that's often what happens.
Yes,
Dosage,
it's different
yeah, and then
dosages.
they give them these crazy names. Who knows who invents these names? Okay.
Yes, and then the Lilly one has two different names too. So I think the Lilly tirzepatide is obviously its chemical name. Then it was as released as a diabetic medication as Manjaro, and then it now is Zepbound, and it's obesity form, and so so those are the main drugs currently driving the market are basically semaglutide and then tirzepatide. But obviously, as with any drug company, they've all got things in the pipeline, and we will talk on new therapies and other things later on in this discussion. If we talk about application as weight loss drugs, obviously this all comes down to that you know that constraint of appetite. Um, I read an article in the Guardian. It must have been like a year ago when I was first really reading up more on this stuff, and it sort of said, one of you lose weight by obviously it reducing your appetite, but actually the person that was writing said it completely removed their interest in food. And I'm like, I don't want anything to do with a drug
Hands
like
down.
that.
Anybody that knows our podcast
Because
has to know this is not something for us. I don't think.
because that's it. It's I think yes, obviously there's a lot of people this is really beneficial for, but it's not something that you enter lightly because it impacts how you think about food and things like that. So yeah,
I know some friends that are on it. It has significantly hindered what they're very happy with the health benefit.
Sure, absolutely.
And I jokingly say it's not for us, but we're fortunate enough that we don't need it. Although, looking at me, you wouldn't be. No, I'm just kidding, John. Right? So, but if you have something that says, "Hey, you need to do this to help," and doctors kind of indicating you'd be maybe better served taking this or helping yourself long term, then yeah. But it definitely has. There's been a noticeable decrease in that. A friend of mine's just sure his appetite, but in general, when he's eating, it's like, yeah, it just doesn't taste the same. I mean, you can
Sure.
still taste it, but it's not the same. And that, but I don't know how that outweighs things, but I hope I don't ever really have to make that decision, right, John?
Yeah, exactly. I hope not too. I mean,
But like you touched
I'm not.
on, you said some interesting thing around like Viagra and that's like a lot of medicines have. This isn't new, right? So
Yeah, yeah.
but like those types of drugs, there's some studies out there that say taking those cardiac medicines, they, John, you and I should be on them right now. Like it'll cut your rate of having a heart attack by like 70%. The numbers are staggering. So
Yeah.
like it is something that I will start to evaluate as I get a little bit older. And it's not for, I don't want to make it into a joke because it's like, oh, you're on this, but like anything that could cut my rate of having something like that happen to me, you should be interested in. The numbers are staggering, John, with that. So this is the same category, right? And potentially we have, it's been proven that these cardiac medications, they say they don't have side effects, but we know there are. And you wonder, but what they're starting to find out over years of researching them is that they are very safe and they have huge benefits, not just with heart attacks. They may be preventing strokes in elderly men, and they're actually thinking about giving it to women. This is like this crossroad with these weight loss drugs. And you can talk about I don't know if the optics on it is what I want to say, but like there's a lot of noticeable people in Hollywood taking it, the scarcity of it, the people who really need it, driving the cost up. There's some things here. And then there is some like, hey, what is the long-term effects of these? It's still a peptide you're injecting into yourself, right?
But, yeah, I mean, I come up with I think all drugs have side effects.
Yeah, well, so, yeah, go ahead.
Do the benefits outweigh the side effects? That's
Clearly
really
they do here. In some, for it's the healthy people taking it that you kind of wonder about. That's what I mean.
So the other guy whose name I've now found is a guy called Rich. Who's the other prize winner, along with Lotte Bjerre-Knudsen, is a guy called Richard DiMarchi, who's now a professor of biochemistry and Guild Chair in Biomolecular Sciences at Indiana University. And like I say, he was at Lilly, and so we lump tirzepatide in with semaglutide simply because it's the two drugs both going at the same market and both showing effect and obviously beneficial effect for people. The tirzepatide is different. It's actually a what they call a GIP-GLP because it actually, if I can find GIP, it's another different approach again. He was working to try and make a different synthetic glucagon to help patients recover faster during hypoglycemic attacks. And he was collaborating with someone called, can't find his name, but they were looking into these different glucagons. One went into obese animals and mouse lost weight, but it was by a different mechanism. And they sort of thought this glucagon be used to generate a futile cycle that decreased body weight by increasing energy use. He wanted to know if this weight loss effect from the stimulating glucagon receptors could be integrated with the appetite suppressing effect of GLP-1, a single safe molecule for unprecedented weight loss. Working with grad students, he found that they could in fact design a single hormone that could stimulate both the glucagon and GLP-1 receptors. And so basically, cut a long story short, they focused on glucose-dependent insulinotropic polypeptide, or GIP. It's another hormone, and like GLP-1, GIP is a gut hormone that triggers insulin secretion. So by tirzepatide actually combines the GIP and GLP-1 mechanism, and it's a GIP-GLP I think GLP inhibitor or something like that. So that's the Lilly drug is slightly different. It's not a pure GLP-1 because it's also got this GIP activity too. And so that's the root of that as the alternative drug.
Yeah, no, it's definitely an interesting story, John. One that's taken it's this isn't an overnight success. A lot of what you just described is going way back into the '90s, right? Some of the earlier type of '90s stuff that I think you're kind of talking about there, like but
Yes.
yeah,
It's
and not just that, like
people, yes.
this is dense. The biology behind this is pretty fascinating. But I think it's important to note a couple. Again, we're back into like finding things from the gut, right? We just touched on that in our news and notes earlier, right? And all of this stuff has blood-brain barrier implications to it. I don't know if you want to talk about that a little bit.
Oh, yes, I was just looking at that. That is it's another fascinating.
Yeah.
These are fascinating
So
drugs.
that becomes a technology. So now that, like, when you think about it, it starts to click as to why there are so many out there and how, you just think, aren't they all the same, or aren't they all just treating diabetes? No, there's so many different things that like we're kind of eliciting, and yeah, there's a lot of crossover, right?
I'll touch on the blood. So basically, it's still, according to this article, how the drugs achieve weight loss has been the subject of discussion and debate. But there was part of it was, could the GLP-1 cross the blood-brain barrier? And so the team at Novo Nordisk looked at liraglutide in the bodies of mice, and while the blood-brain barrier kept liraglutide from moving into the brain, it was able to access small structures that line the brain's fluid-filled cavities, and from there bind to neurons in the hypothalamus, a part of the brain involved in regulating food intake. At the same time, the drug binds to neurons in the hindbrain, her research showed, and activates other circuits. Through these combined effects, the drug affects both satiety and hunger pathways, and effectively stops something called food noise, the relentless desire to keep eating. So it's fascinating.
I mean, it is fascinating, but you can imagine what they just said there is it's just. I'm not saying it's easy to elucidate some of that within a mouse, but it's virtually impossible to do it in a human. Yeah, it just is, and it's great that it's come this far. And it's John, I think it's some with some continued research and continued evolution in biotech and pharma. This might be something that's like in the water, like fluoride, for all you know what I mean? Like, I don't know. Although my son asked me, like, oh, fluoride. I'm like, it's good. He's like, it is? I'm like, yeah, why wouldn't you want fluoride in the water? But, what do I know?
So that's the background to the drugs. Absolutely fascinating in terms of the physiology, etc. Obviously, there's they were developed for type 2 diabetes. They had then the benefits for obesity. That has set up something of a conflict. And also, I think we touched on this a long time ago. There is sort of some medical practitioners don't necessarily view obesity as a genuine condition. It's like, well, you're self.
Yeah, that's important, John.
And so I think there was like the looking at drugs for obesity is almost viewed with suspicion, and it does touch on it again. This article that at Novo Nordisk, they weren't sure they really wanted to go in that direction because is it a real thing? However, let's move on from that. Because you've got the two camps who both want and need this medication, these are obviously fairly complex chemical structures. I don't know how long it takes to make a significant amount of any of these drugs, but right now demand is outstripping supply. So although these drugs are already generating billions of dollars worth of revenue for the companies that sell them, they can't make enough to actually sell as much as people want.
Crazy stuff.
So we touched on the purchase of Catalent by Novo Nordisk a few podcasts ago, because that was like totally out of the blue, and seemed like just a very
bizarre, strange,
a very what's the out there kind of purchase.
But
But
brilliant, right? Like, not I say bizarre until you like, because
yeah,
normally pharma companies or biotechs aren't buying
sure.
CROs, right? But or
But the,
GMP facilities. So it was weird. But then, like when you dug into it, it's like, oh, this is brilliant.
but this is done to help alleviate their constraints in terms of their their supply for semaglutide. So basically, we
I think
go
if you're paying attention, I feel like some other CDMOs or GMP facilities are getting purchased by other
yeah.
giants in this
I
space.
mean,
It's just stuff like
I know,
Domino's, right? That's what's
yeah.
happening.
And Lilly have announced that they're increasing their production capacity, but
Yeah.
right now they're supply constrained up with Manjaro, etc. And so it was just in the news this week. They've increased their guidance for the year in terms of the revenue they expect from Manjaro, but it's still really the supply issue is the biggest challenge for
Mhm.
for these companies.
Yeah.
It's not trying to sell the drug because people want it. It's actually making the drug. So I think maybe we'll go
John, is
on
there?
to
Go ahead. Suddenly I thought, is there challenges to making it? But
the,
let's don't
the,
answer that. There probably are. I
I,
think we'll stick to our agenda. But go ahead.
I would say I don't know enough about it, but I mean I think of I mean
Yeah, we're not GMP guys per se, but
I,
like,
th- I think I
yeah,
think I think the making the peptide is fairly routine, but it then it becomes it's that
it's that
yeah I mean
chain
it,
structure, stability, all that
yeah
stuff. Yeah.
I don't know I would love to know actually, but I
Like
don't.
anything else, after you do it a few times, it might get easier. But I'm sure there's nothing easy about this.
Yeah,
Um,
but it's also who knows. I don't know what the starch material is and how many steps it takes, and that's part
yeah.
of it. Because
No.
even some simple drugs can be an incredible number of steps to get to the end product. I just don't know. I mean, you
Is there
know,
a natural? I wonder if I can get like a natural version of GLP-1, huh? I wonder if that's.
that's where this all started. That's
But I
the
know, but like everything now is, it's a recombinant or however you want to think. It's
sure,
strictly
yeah.
chemistry. It's all synthetic. That's the word I'm looking for.
So
Um,
so we can step on. We can. Shall we?
it's a good segue into the bioanalytical challenges. We started
That's
talking about
exactly where
that.
I was
Yeah, it's
going
a great
next.
segue. There
So,
are
so
some, John, and I
so,
kind of touched on it. I'm talking over you. Go ahead, your turn.
so yeah, if we look at it from the drug company's point of view, their challenge is the supply issue. But obviously, as part of the development of these and new similar drugs, they're still going to be doing PK studies that are going to use labs like KCAS. And like I mentioned earlier on, we have worked on semaglutide and we've looked at liraglutide, um, dulaglutide. We've worked on these drugs are all they all have that they have that peptide chain. They have a lipid addition to it, but the size of the lipid addition it can really have a huge effect on how they behave. So basically on these drugs you've got the peptide, which is really what we would consider a polar um, a polar piece. You add this fatty acid, add this lipid chain. That now you're adding this big non-polar chunks. You completely change the property of the of the of the drug in a bioanalytical setting.
Mm.
If you're looking at a peptide, it's predictable how your chromatography is going to be predictable. You know how you got a good handle on what it's going to do in terms of chromatography, mass spec, etc. Um, these these become chemically more challenged because of that non-polar chain. No longer you're just dealing with a pure peptide. You're dealing with this 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 molecule that has two very different regions that want to behave very differently from each other. And so for us, we found that yes, we can come up with assays. That's fine. But what we found that even though with these changes in length of fatty acid chain, the one assay to the next doesn't necessarily translate because I think tirzepatide it isn't just a pure fatty acid. There's other pieces in it so it will behave differently from others. It's just a pure fatty acid addition. The length of the fatty acid will change how the act within an extraction.
John, so much to unpack with what you're saying. So the first question I have for you.
To keep it simple.
It's great. You're trying your best, but I'm going to ask the first one, right?
Sure.
You're saying that all the drugs on the market themselves, even though there's no, I guess the way I think of it, there's no universal assay for GLP-1 peptides, right? Because of the way or is that a fair statement? Or is there like a off-the-shelf method that we could use?
We have, well, certainly with LCMS, we found that one set of conditions that would run ideally for one don't really translate to another.
But that's not surprising, right? If the fatty acid chain is different on each of them.
Yes, exactly.
And that's my next question, because
it's a huge change.
I'm not a mass spec expert, but I hang out with people that are, right? Yeah. And so there's peptides. It makes sense that a peptide's straightforward because it's got kind of like a sequence to it that is a weight to it that and then maybe it's down an amino acid here or there, so you kind of get a good understanding of where it's supposed to be, right? You put these chains on it. How is each of the chains different? Are there more than one chain, right? How does that?
The current drugs at the moment all have one chain.
Yeah, that's what I figured because of the real estate. I know that, but within that, sorry, John, because I kind of think of like pegylation of stuff, right?
Yeah, that's exactly what it's like. Yes,
does each fatty acid chain have a different? I might be now as a density to it or whatever the size that the spec is looking at, on the reader. Is that so? Does there a profile to, or is it kind of a solid line with just think of it like the analytical method for it versus the bioanalytical method? Talk a little bit about that.
So the way I would look at it is that the one set of conditions that you have for one will give you're aiming for particular sensitivity. That may be good for that one, but if you then apply those same conditions to the next with maybe a longer fatty acid chain, that actually your sensitivity may be garbage in comparison.
Okay.
So you really
have to lot. That's good to know, right? So just because you got a functioning method for one of these GLP-1s doesn't mean you're just going to plug and play and it's easy to adapt to it. It might be, right? It might be, but there is some development and you still got to revalidate the method.
Well, you're going to have to revalidate it because it's a different analyte, but it may give you the direction to go in, but you don't go into this. If I was going with a simple, series of analogues of a small molecule, you'd expect that one set of conditions will work for most of them. There might be something with an bit more outlandish side polar or side chain that affects it, but it doesn't really translate one to the next.
Can I ask? I'm going to take a left turn on you. What about lot-to-lot variation within the drug, the same drug?
I'm not concerned about that.
It's pretty consistent. So even back to that one question, I don't know if you actually answered it. I probably talked right through you, but is within the drug products themselves, is the consistency of that fatty acid chain pretty good, or is there variability in like the size of that chain?
I think as it's made as a chemical, I think it has to be consistent.
Unlike PEG, right? PEGs are messy, right?
I think. Well, pegs, but you're talking, tens of thousands.
Yeah, I guess they're not even comparable, right? So even what I'm saying, even if this is 10% different, the mass spec's not going to see that. Right?
Yeah, these are ones where you've basically got the peptide won't differ from lot to lot, and you're adding another group. And I don't know all the steps to go through all this to make the actual compound, but it's going to be the same batch to batch. If it isn't, you got a problem.
I'm losing track of time. I've suddenly become very interested in GLP-1 bioanalysis, right? So we got one more item to go over.
This GLP. This isn't actually the biggest issue with them. The biggest issue is actually they're a pain in the neck so for us, LCMS is a sequential process. So it's like if you have a batch of 96 samples, say a 96 well plate, you inject sample 1, then inject 2, analyze that, inject well 3, analyze that, and you go through it. The ideal assay, if you have one going from one sample to the next, you see no effect from one sample to the next. It's what we call carryover. You want to see no carryover. That's the ideal methodology. These things, or these analytes, because of that non-polar chain, are sticky. and so what we would call a sticky compound. And so carryover is actually the biggest analytical challenge. That sort of that piece of changing one assay, going from one to the next, that's less of an issue. It's you expect maybe moving from one analyte to another, you're going to change conditions. That we can live with, but that carryover, how we define it is if going from one high concentration to sample to low concentration sample, if you've got a significant amount of carryover, your readout of that low concentration sample is going to be off, because there's a proportion that's come from that high concentration sample that's artificially boosting it, because these things hang around the system. And so I think it's not just, there's various places in the LCMS path where you see carryover, mainly in the injector and on the head of the column, and then sometimes in the tubing between. But the injector's usually easier to address because you change solvents, cleaning cycles, etc., etc. Carryover in the head of the column can be awful to deal with. So these compounds are sticky, and they basically are an issue in terms of the injector. That can you address, but we get carryover from the head of the column. So that adds a lot of
runtime, right? How do you remedy that, John? What's
runtime and really convoluted approaches to address carryover to get that under control. So because what we needed to produce,
when you say convoluted, is that like having to clean the column with something in between?
Yeah, you can either you can go the approach of doing blank samples between each sample, which you wouldn't want to do because that doubles your runtime. Or what we've done is actually do like if you're running a gradient, you get to the end, you then do like a dummy injection that runs through the gradient again, so it effectively washes the column. So it's all adding cycle time that you don't want to be using, because you want to get on to the next sample to analyze. But because of the nature of these compounds, then you have to do it. That's probably the overwhelming thing you have to deal with. And we could, like say, hopefully with the non-GLP team, we can touch on that, and they can talk more about this.
What John's alluding to is we're maybe this is a good segue. We're gonna have our GLP, the people who do this, come on and talk a little bit more about it, because we find this to be incredibly exciting.
Sure.
Okay, John, this is. I'm done grilling you, right? I felt like I put you on the hot seat.
No, it's absolutely fine, because you can tell I find the topic fascinating. One thing I would like to touch on, because I talked about these. I'm really talking about the non-GLP team's work on these GLP-1 analogues at the EBF in 2022. And one thing we did very quickly was, because these have a peptide route, we also asked the hybrid team to look at what kind of sensitivity could we get if we digested these and went for a smaller piece of the amino acid. Because one thing I haven't touched on, these are around 4,000 molecular weight, so it's like I can't remember how long the peptide chain is, but then obviously the fatty acid on top of that. But if we could digest the peptide portion of that, then we could get a specific peptide, or a peptide that's common to multiples. But the nice thing about that approach is you're going back to that route of having an analyte that behaves predictably. It also takes you away from the carryover piece. So we just did a very quick test as a proof of concept. We never took it into matrix. But if we found that if we were asked to work on these where someone really needed ultimate sensitivity, maybe this would be a route we'd go down, rather than trying to measure them intact. Actually go for the smaller piece to see how low we can go, and hopefully then get away from a lot of the analytical issues that we face with these drugs. So there's things
you talked subtly. You mentioned control, and that's a big So even if there is some level of noise, right, as long as it's controllable, that's how we tackle bioanalytical problems, right?
Yes.
So this was great, John. I feel like we want to touch on the future of GLP analogs or new modalities in therapeutic areas.
Let's touch on this, because I think it's really a future. It's future these, and then where anti-obesity is going, because obviously the pharmaceutical industry is there to make money. And so not only are Lilly and Novo Nordisk are out there by themselves at the moment, but there's a lot of people snapping at their heels. But it's interesting because you have researchers looking at Lilly and Novo Nordisk are looking where these drugs are going. Independent researchers looking where they can go. But then you've also got a number of different approaches to tackle obesity. So if we just talk on, let's wrap up semaglutide and tirzepatide in terms of new therapeutic areas. These are just news items within the last few months. We have seen that. Hold on a tick.
Yeah. Are you talking about a presentation from October 4th, right? Or what?
No. I'm actually I'm just saying. Here's some of the news articles about these two drugs. So Lilly plans for Zepbound label expansion into sleep apnea in mid-2024 on the back of a phase 3 win. So there's sleep apnea. There's a different indication.
Huh.
Ozempic and Wegovy have taken over obesity treatment. Can they help with cancer too? So a study found that they appear to reverse the impairments in cancer-fighting natural killer cells seen in people with obesity. This is, like I say, this is all investigative. This is where we might be going.
I see that. This is some good stuff, John.
So then, semaglutide, Wegovy has shown cardiovascular benefit again. This time in a phase 3 step HFpEF trial on top of the expected weight loss. So there's possible cardiovascular benefits. There's possible
NASH,
is another one.
Man. And then smoking and drinking. Wow, John.
The smoking, drinking. Sorry.
So, John, as we get a little bit older, all we need is some Viagra. All we need is some Viagra and a few shots of Wegovy, right? We're good.
There's question: Can GLP-1s help you quit smoking or drinking?
Yeah.
That was the most out there in terms of these articles.
Well, I mean, you think about it; it's got legs, right? Imagine if you could design one that makes the smoke not like disgust you or the drinking not taste good, right? That could really do it, right? Because it seems to play on the senses a little bit, which does make me a little bit. I don't know about nervous is the right way, but like it's just a little bit kind of somewhat mysterious still, right?
Well, I think it comes down to that piece of the study with mice where it went into the pockets of the brain that helped with feeling safe.
How it got in there, right? Like so, John, you actually the myth of the blood-brain barrier, right? We're all now to say nothing got through there. Well, we know that's not true. Just how it's getting through, right?
Is there another area that a differently designed one could go to that would affect other things? I don't know, but I mean, it does tell you what is out there. So just to move on to obesity itself, there are currently 116 assets in phase 1 to 3. It tells you how much attention is being paid to obesity at the moment. And obviously, Lilly and Novo Nordisk have their follow-up drugs. Some of them are
116 trials.
Yes,
is that it? 116.
116 assets.
Assets. Wow.
Yes. So.
That's like assets mean that's compounds, however you want to think of it, as 116 different potential drug candidates.
Yes, exactly.
Wow.
And in terms of some new modalities, one that was in the news was Fractile have a GLP-1 obesity gene therapy, which outdoes semaglutide for weight loss in mice. So very early work, but now gene therapy is looking at this, and then you've got oral drugs where Lilly and Pfizer have both got oral drugs that are designed for this approach. I know certainly there's a oral form of semaglutide. I'm not sure how I'm not sure how that's been administered or what's there to help it be delivered as an oral drug as opposed to the usual injection, but there are oral drugs on the way. In terms of actually anti-obesity drugs in phase 3, this is all according to an IQVIA survey, there's 7 in phase 3. Boehringer and Ingelheim have got a GIP-GLP glucagon receptor agonist. Eli Lilly have 3 GIP-GLP glucagon receptor agonists, so assuming that they'll be either better or complementary to tirzepatide, and then different indications. Europharma laboratories have a combination of two small molecules that's an oral. Novo Nordisk have a semaglutide combination with another similar compound. It's called cagrilinotide. Schwind, who I've never heard of another GLP glucagon receptor agonist. Takeda have a gastric and pancreatic lipase inhibitor, so it's another oral drug. So, and people are always interested in the oral simply because of ease of administration.
Yeah. Compliancy too, I think too, right?
Yes. So it just tells you how much interest there is out there, and like I say, that's why I thought it was a good time to talk about the GLP-1.
I think one of the biggest surprises is just all the indications that it has, John. Right? So it's really cool. Wow.
Exactly, and because it's I think I found a little figure that's pulled off the web that just showed the number of organs that seem to be impacted by GLP-1.
Yeah, and it's all in the food we eat too, right? Yeah.
Yes. So,
John, I think we're good.
We should.
This has been a very fun episode, one that you're obviously well versed in, a little bit more than I am. I hope I was able to contribute a little bit today. I felt on the sidelines, but that's okay, John. No. Where I'm going is let's talk about our weekend. Maybe I don't know about you, but I got some exciting stuff going on.
I absolutely will do it. I just want to wrap up because I think it was good, but also it's a counterbalance to some of the other.
Yeah, no, I know. That's where I was going,
talking, and I nod and say, yeah.
I know what you mean. Like,
and to be honest, so when you say well versed, honestly, a lot of this I've been paying half an eye to it, but I really learned a lot in the last couple of days, just sort of looking at it and then sort of piecing things together in my mind. And I've obviously known about the drugs and as a bioanalytical person, think about the bioanalysis of them.
Yeah, that's why, John, right? This is for both of us. Yeah.
So the actual, most of the time you see, a new indication. Oh, okay, that's nice, type of thing. But but bioanalytically, these are really interesting drugs.
Yeah, they've been over the last few years. You've just learned about it so much because we're doing it, right? Like that's what happened. Yeah, and that's part of why it's so great to work here. You can almost all the things that are coming to us are like the innovator, innovation wise, and not necessarily what's behind us. So that's why this is so near and dear to you now. It's because you're kind of getting into it, and we're learning more and more about it. I think the entire bioanalytical community is learning a lot about it, right? Not just us. Everybody is, because it's just exploded, right?
As a CRO, it's funny. You're in two minds. You want all your assays to go smoothly. You want all the easy compounds, but ultimately you don't really, because you actually really like things like these that are the analytical challenges, because they're the ones you learn from. They're the ones that you actually grow with, and and
they're the ones that save lives too, or benefit mankind too, right?
Yeah. So it's the fascinating because, like I say, sometimes it's really nice to have a drug that just runs day in, day out. If you get the samples, there's going to be no issues. From our perspective, there's no carryover. Sensitivity is fine.
Those are called generics, John, right?
We have some drugs that I've worked on in the past, like every run you did, you knew it was going to pass. It was going to look really clean. All the standards and QCs would be really tight. And you like those once in a while, but ultimately, how do you that's not very interesting to talk to other people about. The interesting ones are the nightmare drugs.
Yeah.
The nightmares from bioanalytical.
Pound of flesh methods, huh, John? Is that what you're saying?
The ones that bioanalytical.
We didn't even talk about immunogenicity. There is that's still something that's a little bit TBD'd here. I do think people do have to do some immunogenicity against it, but I think it's pretty. To me, a lot of this is peptides are in a strange class.
Sure.
You'll know they're neutralizing because drug's just going to stop working. It's already hard enough to try to measure these things. I don't know, measuring antibodies against them is. I do think there's. I don't think it's mandatory though, John. Right? It's in a class that doesn't make it. I don't think they have to do it, but I could be mistaken. I know we.
Yeah, I don't know because I don't think about that.
I feel like it has to do with so it's low risk, and if the risk assessment of the of it pops up somewhere, then you're going to have to take a look at it. I feel like we have not. I know we've looked at anti GLP-1 antibodies for sure, and I know there are you can purchase them, right? That's how some of the IP steps.
Yeah, and that was actually going to be one of my questions in terms of the LBA approach to these, because I didn't.
Yeah, it's just
I
it's tough.
I
It's tough.
I approach these purely as an LCMS person. That's my background, and so I know that we can obviously do them. They obviously, there's successful assays out there. They fragment. They do what needs to be done so we can measure them. It's really interesting. I mean, I'd love to know what Novo Nordisk did with these in the early days, because I know, for example, when it comes to insulin, that's a little bit bigger. But and when
Yeah,
they look,
we've done anti-insulin. Some of the
yeah,
we have. That's what I was referring to. But
yeah, yeah,
John, I think it falls into one of those categories where oftentimes you got to do it maybe early, and then it can stop. Right?
yeah,
There's
yeah.
some, and then immunogenicity is just evolving so much, John. We
Sure.
probably need to have a podcast in and of itself. The preclinical, non-clinical space is definitely changing, and then there are some recent things within the community where they want to really move away from the bridging format that we do often, and the positive control that we rely on. You've heard me talk about this a little bit. They want to move away from that and just do signals. So put
Sure, yeah,
the
absolutely.
drug
Yeah.
down, like a real serological immunogenicity assay, kind of like what you would do for seeing if a vaccine efficacy, kind of using that approach. But that's a whole nother podcast, and it's a really I think it's going to happen, right? As soon as
Yeah.
it's a train that's getting out of the station, but more importantly, it's not a cost thing or an ease thing. This is just better science. So
Sure. Yeah.
that's the piece that becomes really important. But it is a significantly less. We're doing 3 tiers for immunogenicity, and there's some approaches that could be less. So, John, thanks. Great
Yeah,
stuff. Any closing remarks before I tell you about my?
Yeah, I want to quickly wrap on this on the insulin piece, because it would be fascinating to know how Novo looked at this in the early days, because I've obviously there's this sort of peptides and proteins. There's that sort of we can look at them both with the LBA and LCMS, and there's sort of there's a point at which it becomes, up to a point it's LCMS only, and then or mostly LCMS, and then there's the middle ground where it becomes both involved, and then you go large molecule, and it's like LBA is more accepted than LCMS, and this is not me trying to talk one technology over the other. I visited Novo Nordisk years ago. And like I said, that it was really to talk about insulin, but insulin's 5,700 molecular weight, whereas these are 4,000 molecular weight. And
that's a big part of it, right? So
I presented actually some of our LCMS's data from they did insulin glarge and insulin the quicker there's the different acting timeframes on insulins presented to the Novo group, and the data was immaculate from an LCMS perspective. But their take at that point was, we have a really good group for generating antibodies. We don't really feel the need to look at LCMS. And even though the data that I was presenting totally matched the levels you needed to be at via LBA, that was very much their approach. So I'd love to know, knowing that group's mentality, how did they measure semaglutide being a little bit smaller, but being very much an LBA group in terms of how they thought. Obviously, I'm not going to have that conversation, right? But it would. I hope at some point, maybe I do.
Yeah,
someone from Novo who can tell me all about it. But it was just really interesting as a mass spec person walking in it, and really I was never going to get anywhere with them because they had a way that worked, and I'm totally fine with that. It was very interesting for me as more of a chemist and a biologist to sort of be greeted with that kind of reaction. despite the data. Anyway, let's move on. We're
No,
done.
John, thanks for sharing some of that. For me, this is graduation season, John, right? So
Yep.
I've got a son that the next time we have a podcast, he will, I think, pretend, but it depends on how soon it is, but he's a couple weeks away from graduating high school. So, John, this is eye-popping to me. Never have I been so popular in my life. You should see the number of graduation parties. They started last weekend out here, John. I got like 4 or 5. I need like a event planner for my
Sure.
weekends the next few weekends. And even like the day we're having his graduation party, we're going to be running all over the place. I got family coming in town from Boston, from Colorado, from Nebraska. It's going to be a whirlwind for me. But this weekend it kicks off. I think there are, like I said, about 4 of them we got to go to. We still got some things to plan for, but this has suddenly become the month of May will be dedicated to my son's senior year graduation parties. And actually, I don't mind them, John. I get to hang out with a lot of parents that I like. So anyhow, that's what I'll be doing. And boy, is it a great year to be. I've been landscaping and cooking, and got to make some pasta sauce. I might pull out my pizza machine, but all life's going good. And it's a beautiful time of the year out here. But I'll be tied up with that stuff. What say you?
So when's graduation?
It is the 19th of May, Sunday.
Oh, nice. Okay. Yeah. So for us, I was lucky that this is an afternoon recording rather than the morning, because we're in the middle of having our front porch. Our house is 1872 built. The front porch we used to use a lot, but then we noticed it was quite spongy at the front, so the wood wasn't as good as it could be, and we'd been trying to get builders in to do something about it, and just for whatever reason never succeeded. So we actually have been able to engage what they call morning dew builders, or morning dew as you would call them. It's actually it's a young guy who's actually a best friend of my daughter and her husband. He set up his own business, and he does really top quality work. He's done some stuff inside. So this week has basically been the removal of the front porch that we had. So we've been in demolition mode, and then he's got the house patched up ready to, in terms of, because we're expecting rain this weekend, and then he'll go into building mode, and we'll have a completely new front porch on the house. So it's really interesting to watch how it all comes apart, and then getting an insight into like how the house was built. And we bought the house. It had like vinyl siding on it, and so we never knew what was under the vinyl siding. There's always that concern: was the vinyl siding there to hide stuff?
Yeah.
Or was it actually just because it was the fashion at the time? I think he's found it was the fashion at the time.
Yeah. Good deal.
He said what he's seeing is pretty sound underneath. So it's actually that's been dominating the week. So come the afternoons are usually gone because they're just disposing of all the stuff they've broken down.
Well,
this weekend is actually the first weekend my wife has been back in a couple of weeks, because she's been in the UK for her brother's 60th birthday. So we'll actually get to spend some time together. And then obviously that's been really nice, because we did the trip to Venice, etc., which was fantastic. But then I think she was back a day, went off to New York City to do some work there, and then she was back for like another day and then gone. So we barely had any time to connect. So that's going to be our weekend.
You just reminded me that I've been trying to get the house ready. I mentioned people coming in, and like I got sink issues that have been taking up my Sunday evenings. A neighbor has been helping me. I should have just called the plumber. One of those moments where, sure, I might have saved some money, but it's been 3 trips to Menards and still don't have the damn right piece for the sink. Menards is like a Home Depot, John, out here.
Yeah.
So it's been fun. You're making me cringe a little bit, and I threw 60 bags of mulch down over the last, say, 3 weekends as well. This will be the first weekend I'm not throwing 20 bags of mulch on and I laid 200 yards of the bug, the repelling of the weeds. It's all
Sure.
my body was killing me. But I'm sure our audience is like, hey, why don't you wrap this up? So John, I'm good, John. I'm going to say goodbye to our audience. It's been a pleasure doing this podcast with you. I'm looking forward to number 77. We're going to be up to right with our Discovery Mass Spec team, Don. So
okay. Yes, thank you very much. It's been I'm sorry I got a little bit carried away talking, but pharma and bioanalysis is what excites us.
You got to be unapologetic about your passion. Take care.
Okay.
Bye.